Hi, thank you for coming back to my YouTube channel. So in this video, we'll discuss about the basics of the gene cloning, the introduction of the gene cloning technology. So the first thing is what is a gene cloning. So in order to understand what is a gene cloning, you'll have to understand that what is genetic engineering. Basically, genetic engineering is a process of gene cloning into new organism and altering the gene sequence to change the protein product. So that means you make copies of a particular gene outside, and then you kind of do manipulation to check its functions, etc. So what's a clone? A clone is an exact copy of an organism, organ, single cell, or molecule. Basically, it's a product of asexual reproduction. So that's why it's an exact copy. Now, gene cloning is a common practice in molecular biology lab. So it is used to create copies of a particular gene for downstream application. So what are the downstream applications? That means gene sequencing, if you want to perform some kind of site-directed mutagenesis, genotyping, or a kind of uh, protein expression and protein purification. So the traditional technique for gene cloning involves the transfer of a gene fragment from one organism to a self-replicating genetic element such as bacterial plasmid. And you know that nowadays lots of other host organisms are available. For example, yeast, fungi, and then you have plants, etc. The technique is used to characterize a gene and protein. Now let us come to the steps of the gene cloning. So here you will see that you have a plasmid here. So you isolate the plasmid from a suitable host. In this case, this is bacterium. So this is your plasmid, and then you have isolated. First you identify the gene of interest. So this is the gene of interest. That means this is what you are going to clone. So this gene of interest you isolate from a suitable source. For example, this could be a mammalian cell, this could be any other cell. So now you take the gene out and put it in this plasmid. So this is how you make the recombinant construct. So now once you make the recombinant construct, you put it into a suitable host. You see in this case this is a recombinant bacterium. Now you differentiate between the bacteria which contains your recombinant construct with other bacteria that do not. So in this step is performed by growing the bacteria in a suitable media containing appropriate antibiotic. So the bacteria that harbors your gene of interest or the construct will be able to successfully grow. However, the bacteria that do not harbor your recombinant construct, that will be killed because of the antibiotic. And in the next step you grow them and make bigger culture, and then you can sequence the DNA, you can express the protein, you can characterize its function, so on and so forth. So in brief, gene cloning is a technique using which you take a particular gene of interest out and then you put it in a suitable system, you allow it to grow in a suitable host, and then you perform various characterization. Now little bit of history of development of genetic engineering. So this history could be essential for your competitive exam and knowledge. In 1903, W. Sutton proposed the idea of gene resides on chromosome. It was not known earlier. In 1922, Morgan and his colleagues performed the gene mapping. So they analyzed the relative positions of 2000 genes on fourth chromosome of Drosophila. So please notice that they analyzed the relative position, not the actual position. During 1944 to 52, Avery, McCarthy, and Hersey and Chase independently showed that DNA is a genetic material. And during 1952 to 1966, the structure of DNA came up by Watson and Crick, as you know, and then the genetic code was known, and then the process of transcription and translation came up. In 52 to 66, all this happened. During 1971 to 73, the era of recombinant DNA technology started, and Kary Mullis in 1985 invented the principle of polymerase chain reaction, now famously known as PCR, and that led him to receive the Nobel Prize. In 2000, the human DNA was sequenced by launching of the Human Gene Mapping Project. Now let us move to the steps of gene cloning in brief. So in the first, what you do is that you construct recombinant DNA molecule by taking your gene of interest out and identifying a suitable vector. You put them together in presence of a set of enzymes. So later you'll come to know what are the enzymes required for performing this step. In the second step, you introduce this recombinant DNA molecule or the construct, now called construct, into a suitable host. Here in this case the suitable host is a bacterium. Now the bacteria harbors your recombinant DNA molecule containing a gene of interest, and in the next step you differentiate or isolate between the recombinant and non-recombinant bacteria in the presence of a suitable antibiotic. So the recombinant bacteria are of your interest, and finally what you do, you allow this recombinant bacteria to grow in a large scale, and then you can perform kind of purify the DNA molecule. You can allow the protein to express by that particular gene, and so on and so forth. So now the question comes that what is the importance of gene cloning? So the foremost important task is that once a gene is cloned, there is no limit to the information that can be obtained about its structure, expression, and function. So together this is called characterization of a gene. So if you want to characterize a particular gene which was not known previously, so the gene cloning is essential. And the second is that the availability of the cloned material has stimulated the development of analytical methods for studying the genes, with new techniques being introduced all the time. So this is a passive importance of the gene cloning. I hope that you understand the basics of gene cloning up to this. Please bear with me for more videos in this series. Please subscribe my channel for getting the new videos in this series and important videos of other sections of life science and biology. Thank you.